Learn the main lines and key ideas for how should black contest the white pawn center? There's a better move. Try again and look for a strong pawn break. Good idea. What's next? Only one move now. Well played. It may be surprising, but black has no trouble after the queen trade. In the endgame, the right to castle isn't such a serious issue. How should black occupy the center and prevent white from playing a quick e2, e4? White targets b7 and d5 with a good position. Try again and look for a stronger way to occupy the center. Black can meet d takes c5, with queen takes d8. White wins a pawn. Try again and look for a stronger move to occupy the center. There's no need to give up the bishop pair. Try again and look for a strong... Good idea. Black will keep a piece on e4 to prevent white from gaining it. Again, white is ready to play e2, e4 and dominate the center. How can black fight against that plan? Great idea. White's pawn on f3 makes much less... How should white win material?
When white opens the game with the king to explore, but another solid choice is to simply play e6. Black's idea is to strike against white's center with moves such as c5 and knight c6, when black shouldn't face any serious, not a challenging option at this. King can solidify the center with bishop, preparing the pawn break c5. A pot is the short veer for dealing the dark squared bishop. Black should not as white. Adding Former world champion Vishwanathan against the D plan is knight g to e7, preparing to play bishop g4, pinning the knight on f3, followed by knight f5. Played bishop, released the tension, sets up a small trap, takes d4. Not ignore, as this allows white to exploit black's exposed king, an undeveloped kingside, knight d7. The knight without giving up the d5 pawn, opening up the game position. Changed a pair of minor pieces with knight takes d4. Bishop takes d4. Move the knight game, helping white superior d with ignore white's serious lead in development and release the tension with the opposite pawn with knight takes e5, as this allows white to exploit black's urging black to move the knight. Black needs to activate this with knight takes d4. Bishop takes d4. Black needs to activate the king side in the e5. This was going to play e7 until to comfortable d takes c6. White pinned the knight e7, comfortable position. If white tried to discourage black from castling with queen, ignore white's bishop. Solution is queen c7, 5 killing. White may decide while adding additional pressure against this with game. Rather than castling, white may decide to immediately support the center with bishop e3. Protects a position to b6, knight c3 pawn. By the surprising queen b1, e2, attacking the queen, followed by queen takes b7, attacking the unprotected rook on a8. 1. White enjoys a significant lead in development while the b7 and c playing black. His opponent takes b7 or world champion. Challenger Sergey to turn this active position into a fine victory. Since queen takes b2 allows white the type of active chances the short variation tries to produce, a more reliable option for black is to play knight c6. Continuing to intensify the central struggle. One popular option to explore is kingside castling, but it's also important to understand bishop takes c5. It looks like white is simply helping black catch up in development, but this is like c bishop. Grandmaster Shakri Grandmaster encounter, Hikaru Nakamura played knight b5, threatening both knight c7 and knight d6 check. So his opponent, Grandmaster Shakhriyar Mamedyarov, played the prophylactic king f8. Black's king is immune from any annoying knight checks, and knight d6 would not be a wise decision, as the e5 pawn would come under serious pressure, and with it, the knight on d6 would become quite unstable. White will favor centralizing the knight with knight b to d4, followed by kingside castling. White enjoys a central space advantage, although this is still double-edged as it will serve as a target for black. Meanwhile, black will likely continue with knight g to e7, and one idea is to expand on the kingside light squares with h5 and g6, taking space and preparing to move the king to g7. Since the center remains closed, black's king should not face any immediate problems. Slightly strange Laro Khan. Knight f3 is an Grandmaster tried a game match and is regularly natural continuing 4. The light squared bishop can try to run with bishop e8, bishop g6. When white piece, build a space advantage on the king side with its tracks with h and h5. This is to add no longer a threat, bearing kingside development 3. Pawn break, the approach may come under should consider the possibility of queen b6. Adding pressure against the white can control of the pawn, canceling. This being in mind, but another on the c5 both ways to change pawns and 6. Exchanging the bishop square. His 6. Knight f3. This allows black to so continue. See knight out for b6 with bishop takes range for strong control. Bishop immediately activated the light b3. Petrosian wasn't comfortable with the placement of white's queen and decided to pawn at the expense of misplacing black's knight on the edge of the board. Has retreated, Bob's position, and although black is certainly doing okay, his light-squared bishop is less active than black would like, and the wayward Fischer went on to win. The able choice is to play white would, so a sensible choice with developing the queen knight d6, unpleasant knight advance to e5, so former world e6, hit square, preparing to castle, played bishop d6, protecting the e5 square, preparing to castle, and offering an exchange of dark squared bishops. His opponent, Grandmaster Levon Aronian, accepted this offer with bishop takes d6, queen takes d6. Exchanging the dark squared bishops involves a potential pawn sacrifice, allowing white to play queen takes b7. Of course, white does not have to accept this sacrifice and may decide to castle, following the simple, safety-first attitude of the exchange variation. but Grandmaster Aronian challenged black to prove compensation after queen takes b7. Although white has captured a pawn, this does open up the b-file, allowing black to generate counterplay with rook b8, forcing the queen to vacate the b-file with queen a6, when black secured the back rank by castling. Black may be down a pawn but enjoys a lead in development with an active position. He's prepared to gain a tempo against the queen with rook b6, followed by doubling rooks with rook f to b8, creating serious pressure down the b-file. Black has full compensation for the pawn, and even managed immediate pressure against d5. Black still continues with knight c to support d3 pawn center with 7 and 4. Constantly advertising he takes the d pressure against pawn b7 will king signal. Like many, he must be calculating, but the is for counter. Grandmaster Giri supports white must play on while attacking the 4, provoking the forcing sequence bishop b5 check. Knight takes b5 allows 6 check, king e7 takes b. Although black's king doesn't look especially healthy on e7, it isn't so easy for white to directly exploit it. His position and followed by bishop g5 check, leading to simplifications, proper play of a position. This variation will typically reward the better prepared and more experienced player. We have spent most of our time focusing on white responding to response defense, vulnerable to a one-side pawn and showing to f typically play nothing wrong, but if you're interested in g to f, hoping from cap, knight takes, provoking bishop d2, in order to add pressure against the d4 and b2 pawns by the queen. 3. Tied down to the king Slov system with knight d7 is definitely worth considering. Pawn black's bishop on obligated takes g6. This with h4 stable chore, attacker. 3. H5 and a scalar is nothing wrong, eyeing the e5 square. So black can develop side exclusively opened agent, white on h and attempt black e6 solid carol f7, that white black struck between the d5 7, controlling the h2
Chị đã bán này có đi xem nhá. Đúng không?
Good job building a lead and not letting up. Knight develops. Their best option was to prepare a bishop. Nice, their best option. This is a clear. They've targeted. Your best move was to develop a knight off its start. They've targeted your moving their threat. Clearly, you don't. Their better move was to force. That was a clear recap. That was an even, a straight. Your better option was to force a straightforward recap. Your better. They've brought their piece into play. For sure, you had to. Their better. This move active. They played opposites. They are. You prevented your opponent from being able. They exchanged. You win material, taking advantage of the pinned defender. A clear recapture. You captured a takes. The only move. They defend. Yes, defending. Your rook takes. That's playable. Your best move was to prepare a retreat square. Their best option was to activate a bishop. Your doubled rooks work great together. They made one of the top moves in this position. This is the way to win. Decent move, but they had a. This wins a pawn.
Algebra's like sheet music. The important thing isn't can you read music, it's can you hear it.
Carolyn Bertozzi was born 1966 in Boston, Massachusetts, USA. She was awarded a PhD from the University of California at Berkeley in 1993, and is now active as the Ann T. and Robert M. Bass Professor at Stanford University, where she is also an investigator of the Howard Hughes Medical Institute. So by that, please please join me in welcoming Dr. Bertozzi up on stage.
Thank you. This is the honor of a lifetime, to say the least. And it's my great pleasure to tell you about the work that we did to develop bioorthogonal chemistry and to apply this new chemistry to imaging of cell surface glycans and many other applications related to that. Let me start by just introducing one of the basic tenets of the field of chemical biology, which is my field. And one of our tenets is that there are many unmet needs in the biological sciences and in medicine that drive the development of chemical technologies. And bioorthogonal chemistry is very much one of those technologies that was inspired by an unmet need in biology. And at a very high level, bioorthogonal chemistry was developed by my coworkers and myself when I started my career at UC Berkeley. And the fundamental goal was to invent a new type of chemistry that could be performed in what I think of as the most complex reaction vessel you could think of, which is the living organism. And this flies in the face of the traditional ways in which we think about chemical reactions. When we do reactions in our chemistry laboratories, we're typically using this kind of a reaction vessel. It's a round bottom flask where the chemist can control every single parameter of that reaction. By contrast, if your goal is to do chemistry in a living system, as was our goal, you're no longer in the reaction flask, and you no longer have control over all of the parameters. Instead, you might be doing your chemistry in a live cell, in a cell culture dish, for example. And there are a lot of interesting problems you could solve by doing chemistry in the live cell. For example, to attach imaging probes to biological molecules that you want to study in their native habitats. And that's a very complex environment in which to think about chemistry. But even more complicated is the chemistry you might want to do in living organisms, like a laboratory mouse, or, as I'll show later in this talk, the zebrafish, which is a model for development of vertebrates. And probably the highest level of complexity, where one might want to do a chemical reaction, is the human. And in fact, as you'll see, bioorthogonal chemistry is actually now being tested in human cancer patients for the purposes of drug delivery. So for all of these kinds of reaction vessels, we knew that we needed a different brand of chemistry. And so we coined this term bioorthogonal to describe the qualities that you would need to do chemistry in these living systems. So our definition is that bioorthogonal reactions are reactions among components that neither interact with nor interfere with a biological system. And one way to think about that is that there is an enormous reactivity space that's described by biology. I like to think of biological reactivity space as a galaxy of chemicals within a much larger universe of chemicals. And it's a large galaxy, but there's a boundary around it. And somewhere outside of that biological reactivity space is the bioorthogonal reactivity space, a different galaxy with different chemicals, functional groups that you would not find in a biological system, but that will react with each other regardless of being surrounded by all of those biological functionalities. So that's the type of chemistry we'll be talking about today. Now, as I mentioned, chemical biologists often develop new technologies in response to an unmet need in biology. And in my lab, the unmet need that we became interested in was to study changes in cell surface glycosylation. That's another word for the complex carbohydrates that decorate the surface of all of our cells. And even back in the 1990s, there were decades of literature showing that when cells undergo a transformation of their state, there are changes in the patterns of cell surface glycans. And sometimes those changes in cell surface glycosylation are associated with disease. For example, it was already known back then that when healthy cells transform into a cancerous state in the context of a tumor, there are cell surface glycosylation changes that often include an overproduction of a particular sugar that's known as sialic acid. And it's one of the building blocks in the complex carbohydrates on the surface of your cells. And we had a fairly straightforward objective, which was to develop a technology that would allow us to profile the abundance of sialic acids on healthy cells compared to cancer cells, and to develop a strategy for molecular imaging of changes in sialic acid in order to study cancer. But at that time, there was no technology available to image cell surface sugars, including cell surface sialic acids. So that was the unmet need that we were focused on solving. How do you image a sugar? Well, you have to do it in a different way than you would image other biomolecules, like proteins or nucleic acids. For those other molecules, there were some good technologies available at the time, but for sugars, there was not. And during my postdoctoral years, so this takes me now back to around 1991 or 1992, I was thinking about this problem, the need to image the cell surface sugars. And I happened to attend a conference in Southampton, England. And this was a serendipitous moment for me because I heard a lecture by the late Professor Werner Reuter, a very well-known German biochemist at Free University Berlin. And his lab had been studying the biosynthetic pathway that produces sialic acids and delivers them to the cell surface. And what he had observed, and he presented this at this conference, is that one could feed cells a chemically altered version of a metabolic precursor of sialic acid that's called N-acetylmanosamine. And normally, N-acetylmanosamine has an acetyl substituent here at this pink position, where this R group would be a methyl group. And what he found is that one could make conservative changes to this R group, just extend the side chain out by a few carbon atoms. And those subtly altered N-acetylmanosamine derivatives would be subject to enzymatic metabolism inside the cell, which led to the production of an altered sialic acid residue, which was then incorporated into the complex carbohydrates on the surface of those cells. So you feed the cell a chemically altered sugar, and they produce a chemically altered sialic acid. And when I saw that talk, a thought came to mind, which was that maybe we could put a chemical group that's reactive at that position. And once that chemically reactive group appears on the surface of the cell, we could chemically attach an imaging probe. So that was one of the research proposals that I crafted and submitted when I applied for a faculty position at UC Berkeley. And here's a cartoon that's almost literally right from my job application. from 1994, where I proposed a two-step process for imaging cell surface glycans. The first step would be to feed the cell or the organism, or maybe the human patient someday, that chemically altered simple precursor sugar. And that sugar would be metabolized by the cell, converted to the cell surface product, where this chemical group X, and I was agnostic about X at that time because I wasn't quite sure what X would be, but some chemical group X would come along for the ride. Now it's presented on the cell surface, and we would do a chemical reaction with an imaging probe that had a complementary functional group Y. And the hope was that X and Y would react with each other, form a bond, and that would attach the probe to the sugar, and we would now be able to image the sugar. But there was one problem with this cartoon, which is that we would need a chemical reaction between two functional groups that could proceed in a living system, in cells, in animals, the natural environment of these cell surface sugars, which means that the chemical reaction would have to be bioorthogonal. And this was the problem that was the genesis of bioorthogonal chemistry. So the reaction that we started with, and which ultimately was the first bioorthogonal chemical reaction, was a derivative of a very well-known chemistry from the German literature of the previous century. This is the classic reaction of triphenylphosphine with what turned out to be a very important functional group. You'll hear a lot more about this in the other talks this morning, which is the azide. And phosphines and azides will react with each other to form an adduct called an azaylide. This was a chemistry first reported by Hermann Staudinger over 100 years ago, 1919. And in fact, Professor Staudinger won the Nobel Prize in Chemistry about 40 years after this initial report. Now, phosphines and azides do not exist in biological systems, and they react with each other very selectively without any interference by the biological system. So on its face, this reaction, as I've shown it, looks like it could be a bioorthogonal chemistry. But there was one liability with this classic Staudinger reaction, which is that the azaylide product is not stable in water. And if you're doing reactions in living systems, water is your solvent. There's no choice. So we had to solve this problem of the hydrolytic sensitivity of the azaylide. And one of my graduate students, Eliana Saxon, who joined my lab around 1998, came up with the solution to the problem. She made a simple modification of triphenylphosphine, introducing this methyl ester. So now, when the phosphine reacts with the azide to form this azaylide, the nitrogen atom, which is nucleophilic, can attack the ester in an intramolecular reaction, very rapidly forming this cyclic product. So now, when water ultimately hydrolyzes this phosphorus-nitrogen bond, the amide bond that had formed in that intramolecular reaction keeps this product together as a ligation product. So this variation on the classic Staudinger reaction, we called the Staudinger ligation. And this was the first example of a bioorthogonal reaction. And here is Eliana. I found this old photograph. The day that she got her first big result, where she showed the Staudinger ligation could be performed on cultured cells in a dish with no harm to those cells. And she came into my office and she showed me the data. And I remember thinking, I think this is a really big result. But this was very early in the days of my young lab. So once we had this chemistry worked out, we then demonstrated that we could introduce the azide into a variety of different cell surface sugars, all of which are interesting in their own ways. So for example, I mentioned that imaging sialic acid was one of our initial goals. And so we showed that we could introduce the azide on the side chain of N-acetylminosamine. And when fed to cells and metabolized inside the cell, the corresponding azidosialic acid would appear on the cell surface, primed for reaction by the Staudinger ligation. And likewise, we figured out how to put an azide into a sugar called fucose, which is also an important sugar for various biological systems. And in addition, we showed that we could put azides into N-acetylgalactosamine, or GalNAc, using this azidoacetyl variant of the parent sugar. And each of these sugars we chose because there were some interesting biological stories behind them. Another big moment in the lab came when two of my former graduate students, Jen Prescher and Danielle Dubey, demonstrated that the Staudinger ligation could be used to attach probes to sugars in living mice. This was another big moment in the lab because it was the first demonstration that a bioorthogonal reaction could be so well tolerated by a living system that you could do the chemistry in the mouse with no toxic side effects. And in fact, Jen and Danielle, I'm so privileged to say, are with us in the audience today. And here is an image from this paper they published showing that they could inject into mice azidoacetylminosamine. It would get metabolized by the cells in the body of the animal, and the azidosialic acid would appear on the surface of those cells. Then, in a second step, they would introduce a phosphine probe set up for the Staudinger ligation. And in the body of the animal, the phosphine would find the azide, undergo the reaction, and form this product, which we could then detect in a variety of ways, including with fluorescent tagged monoclonal antibodies. Another major achievement by these fearless students who were willing to turn the mouse into a reaction flask. But we knew that there was more work we had to do in reaction development. And the reason was that the Staudinger ligation, for all of its amazing qualities, was a rather slow reaction at biological temperatures. It's a bimolecular reaction, which means that it follows bimolecular reaction kinetics. And so hopefully the students in the audience will recognize that for such a reaction, the rate is equal to an intrinsic rate constant times the concentration of the two reactive partners. And in a biological system, like a living animal, we learned some things that are important. For example, we learned that the concentrations of these reactants were probably going to be very low. The animal is metabolizing these reagents as soon as we put them in the body. This is not a reaction flask in your fume hood. And there's only so much of a molecule you can introduce into an animal because of issues like solubility in aqueous systems or non-specific toxicities for molecules that really don't belong in the animal's body. So given the limits on the concentrations of these two parameters, the intrinsic rate constant of the reaction is very important if you want the reaction to proceed at a reasonable rate. And for the Staudinger ligation, which had a second order rate constant around 10 to the minus 3 per molar per second, we discovered that this was a little too slow. This reaction took hours to proceed, sometimes a day. And there's a lot of biology that takes place on a much shorter time scale than a whole day. So we were interested in alternative bioorthogonal reactions that would leverage the azide as a reactive partner. Now it turns out that azides can also undergo what we call 1,3 dipolar cycloadditions with alkynes. This is another chemistry that was well established in the previous century. And Professor Rolf Huisken from University of Munich was one of the major practitioners of this chemistry, who did a lot of mechanistic work to describe how this reaction proceeds. And like the classic Staudinger reaction, This chemistry seemed to us as being almost bioorthogonal because, like the azide, alkynes are also quite rare in biological systems. We don't have them in our bodies, as far as we know. And yet these two will react with each other to form a product called the triazole. Now, the reason I say this is almost, but not quite, bioorthogonal is because this reaction is very slow at room temperature, much slower even than the Staudinger chemistry. Which is why when people do these reactions in the fume hood, in the chemistry lab, they generally have to reflux the reaction at high temperatures, like 100 degrees C. So at biological temperatures, we thought of this Huisken chemistry as just being too slow for our needs. Now, in 2002, the world was changed. And that's because my co-laureates, Professor Barry Sharpless and Professor Morten Meldal, discovered a mechanism for accelerating the rate of the azide-alkyne cycloaddition. And this is what is now almost synonymous with the term click chemistry. And as you will hear from their glorious lectures after mine, the addition of a copper catalyst changes everything. And suddenly, this reaction is incredibly fast. It's over in minutes. And it is now by far one of the most important reactions in all of chemical biology, and very much worth recognition by a Nobel Prize. Could this solve our problem of kinetics? And the answer was not quite. The problem was the copper catalyst was just too toxic for cells and live animals. But it was kind of on the right path, and it got us thinking about other mechanisms by which one might accelerate the rate of the cycloaddition, but without the need for a toxic metal catalyst. Now, like many of us, I teach sophomore organic chemistry, introductory organic chemistry. And when we teach this class, we always have a lecture on the concept of ring strain. And here are the two textbooks that I used to use to teach this class when I was at Berkeley, both written by Berkeley faculty, Peter Volhart, Clayton Hethcock, and Andy Streitweiser.
Hello, thầy Trọng xin chào các em. Hôm nay chúng ta sẽ cùng thầy
Toàn nó nghe cũng rõ rồi gì em. Thì cho tầm soạn ở đâu có rồi trên mạng hay nào hôm nào cũng đã này. Thứ nhất là điểm cao này, thứ hai nữa là muốn đi quá nhiều, quá sâu, quá lan man gì cả, sẽ tập trung vào những phần quan trọng nhất. Sẽ yêu cầu các em phải hiểu bài chứ không. Thế thì các em sẽ có vào phòng thi thì các em sẽ bác thăm ba cái câu hỏi thuộc 3 phần khác nhau, có nghĩa là mỗi phần một câu. Cô sẽ giúp bạn ấy, cô sẽ phần đại cương. Để tóm lại là cái phần này á, cô cũng nói trước là nếu mà ngồi học thì nhiều khi cũng cảm thấy nó có thể hơi khó hoặc là cảm thấy hơi chán, bởi vì nó tương đối là nhiều khái niệm mới, hoặc là hồi đấy là các thầy học ở Nga về biên soạn thì cô giáo có thể đây để có khác thầy học ở Nga về biên soạn trước, cho nên là nó sẽ theo một cái logic khác. Để cô giáo xem nào, để cô giáo có thể đây để các cô sẽ thì trong cái 3 cái quyển mà hóa cơ ở trong giáo trình ấy, thì nó hơi khác một chút, có nghĩa là gì? các thầy sẽ tập trung có viết một cuốn là chuyên về mạch hở. Đấy. Một cái phần cuốn là chuyên về mạch vòng. Như vậy quyển 2, quyển hóa hữu cơ 2 là chuyên về mạch hở, quyển 3 là chuyên về mạch vòng. Như vậy là nó sẽ hơi khác với cái logic mà cô đang dạy học ở đây. Cô dạy thì sẽ các chất theo nhóm trước, có nghĩa là dạy từ đơn giản đến phức tạp, đúng không ạ? Từ các hydrocarbon này, rồi đến ancol, rồi đến aldehyde. Nói chung là theo cái logic như phổ thông vậy ạ. Thế còn các thầy thì lại dạy theo cái mạch carbon, có nghĩa là mạch hở thì dạy ở phần riêng, sau đấy ý là sau đây thì cô giáo sẽ đi vào cái phần chương đầu tiên, đó là chương đại cương về hóa học hữu cơ. Trong cái chương này thì chúng ta sẽ cần phải nắm được những cái gì? Đó là nắm được cái thế nào là đồng đẳng, đồng phân này, đúng không ạ? Đó là các khái niệm thôi ạ. Các em đồng phân hình học, đồng phân quang học. Đồng phân quang học là phần mới. Tiếp theo là các cái khái niệm để ôn lại như là nhóm định trước này, bậc nguyên tử carbon này, vân. Rồi học về các cái tác nhân phản ứng hóa học, các kiểu cơ chế phản ứng. Rồi cô giáo sẽ giới thiệu cho các bạn một số cái phương pháp vật lý tách tinh chế, xác định thành phần và các hợp chất hữu cơ. Đây là những cái thì hợp chất hữu cơ thì có một cái đặc điểm là như thế này. Nó là các hợp chất bốn nguyên tố carbon, đúng không? Chúng ta biết rằng các hợp chất vô cơ thì rất là nhiều, vô cơ thì nó rất là nhiều nguyên tố, rất là nhiều nguyên tố, nhưng mà hợp chất hữu cơ thì nó chỉ có một vài nguyên tố thôi. Ví dụ như là carbon này, hydro này, oxy, nitơ, lưu huỳnh, phốt pho, còn có thể có một vài cái kim loại. Đấy, kèm nữa đó là hợp chất cơ kim ấy. Tuy nhiên là cái hợp chất hữu cơ thì nó có một cái đặc điểm là vì nó có khả năng là nó tổ hợp, đúng không ạ? Từ vài nguyên tố nhưng mà nó có sự tổ hợp rất là đa dạng, cho nên là nó có số lượng các cái hợp chất hữu cơ là rất là nhiều, và hợp chất vô cơ chỉ là số lẻ so với cái hợp chất hữu cơ thôi. Thì hợp chất hữu cơ thì nó cũng có ứng dụng khá là đa dạng trong thực tế. Và hiện nay thì mỗi một năm người ta cũng đã phát hiện ra rất nhiều các hợp chất hữu cơ mới. Bản thân cô khi mà làm tách chiết các hợp chất hữu cơ thì cũng đã tách chiết được khá nhiều hợp chất mới và đăng được một số bài báo trên tạp chí quốc tế. Thế thì hàng năm người ta cũng khám phá ra hoặc là tách ra được rất nhiều, hoặc là tổng hợp lên rất nhiều các hợp chất hữu cơ mới.
Con học xong 10 á.
Là tốt nghiệp thì cũng bạn ấy cũng thì bản chất của hóa dược là hóa hữu cơ, có nghĩa là bản thân những đồng nghiệp của cô mà đã từng làm việc ở nước ngoài, các bạn cũng sau khi tốt nghiệp thì bạn ấy cũng vào các công ty dược làm chính. Đấy, thế thì bản chất của cái tổng hợp hóa dược ấy thì chính là hóa hữu cơ thôi. Thì đấy là ứng dụng trong dược phẩm, tức là tổng hợp với các loại thuốc. Các em mà có điều kiện mà có thể thử lên mạng mà tra được, ví dụ như là tổng hợp các cái thuốc như Tamiflu chẳng hạn, tổng hợp cả nhiều người để hạn cơ hợp cái tốc độ phản ứng bị oxy hóa, bị hỏng thuốc. Đấy, nó rất là kém bền với không khí, về mế nhiệt. Đấy, nên là dễ bị oxy hóa, đúng không ạ? Và dễ cháy. Cái điểm dễ cháy là vô cùng quan trọng. Ví dụ sau này các em học hóa dầu hoặc là các bạn làm về các cái lĩnh vực có liên quan dung môi thì cái việc mà cái điểm dễ cháy này là hết sức quan trọng. Ví dụ như các em làm thì các bạn học hóa máy hoặc là bạn học quá trình thiết bị mà các em đi cần có một cái đơn đặt hàng để chế tạo các cái thiết bị cho một cái nhà máy cồn chẳng hạn, thì tất cả các cái động cơ cũng là phải là động cơ phòng nổ, đúng không ạ? Đấy, động cơ chống cháy nổ. Đấy là để tránh việc tạo ra tia lửa điện để mà có thể làm cháy nổ cái dung môi. Đấy là điểm rất là quan trọng khi mà các em làm việc với cái đối tượng nào thì các em phải tìm hiểu cái đối tượng đấy. Đấy là cái điểm dễ cháy là vô cùng quan trọng. Đấy. Thì khi mà làm việc với các chất dễ cháy như vậy thì cái việc mà các em ở trong nhà máy chẳng hạn, các em có cái việc mà bảo hộ cũng như là về phòng cháy chữa cháy là hết sức cần thiết và là bắt buộc. Đấy, ví dụ làm một cái phân xưởng gì đấy mà có người sản xuất hóa dược hoặc là sản xuất liên quan đến cồn rượu chẳng hạn, hương môi chẳng hạn. Đấy thì chúng ta đều phải có hệ thống phòng cháy rất là nghiêm ngặt. Chỉ cần một anh nào mà công nhân hút thuốc ở trong đấy có khi là đi luôn cả nhà máy cồn. Đấy, thế là rất là quan trọng cái việc là nó là chất dễ cháy. Điểm tiếp theo đó là các hợp chất hữu cơ này thì nó có một đặc điểm là thường là ít tan ở trong nước. Đấy là đối với những chất khô phân cực, ví dụ như là chúng ta thấy xăng dầu chẳng hạn thì chúng ta thấy là nó nổi lên ở trên nước, đúng không ạ? Đấy là những chất ít tan trong nước và nó sẽ tan nhiều hơn trong các dung môi hữu cơ. Cái điểm này thì nói chung là cũng tùy thôi. Thường ví dụ có khó rõ chức nữa các khẩu anh thường chọn được như là benzen, thế còn gì
đồng đẳng là cô đợi em chút ạ. Những chất hữu cơ có thành phần nguyên tử hơn kém nhau một hoặc nhiều nhóm nguyên tử nhất định ạ. Nhưng có cấu trúc hóa học thì tương tự nhau. Có cấu trúc
cả. Thứ nhất là đồng đẳng. Đồng đẳng là gì? Đó là những hợp chất hữu cơ có thành phần hơn kém nhau một hay nhiều nhóm nguyên tử. Ví dụ như là chúng ta học dãy đồng đẳng của ancol, đúng không ạ? Đấy, thì chúng ta có metanol này, ethanol. Đấy, thế thì những cái hợp chất này thì nó có một cái đặc điểm là nó sẽ có cái phần nhóm chức là rất là giống nhau, đều có nhóm chức OH cả, cho nên là tính chất hóa học của nó là tương tự nhau, nó gọi là đồng đẳng. Các hợp chất này nó sẽ hợp lại thành một cái dãy đồng đẳng, đúng không? Tuy nhiên cũng phải nói với các bạn một chút là các cái hợp chất mà ở đầu dãy đồng đẳng thì nó sẽ có cái tính chất nó hơi khác biệt một chút so với không có phản ứng. Nói đó là những hợp chất mà có cùng công thức phân tử nhưng cấu trúc hóa học khác nhau, đúng không ạ? Chính vì cấu trúc khác nhau cho nên là tính chất hóa học nó cũng khác nhau, đúng không ạ? Cứng nhắc và E này, Z là E này xuất phát từ tiếng Đức là đấy. Thế thì người ta lại thấy ra sang hai cái số 6 này số 5 này phát hiện ra rằng là nó là carbon trên này là các cái nguyên tử hai nhóm có tính không trùng vận ảnh. Thì sao em? Góc 180 độ, đúng, không có tính. Em chuyển file đi orbital P đấy, đúng không ạ? Và orbital P đấy là cái orbital được thêm một electron ạ. Hiệu ứng cảm ứng dương ấy, thì sẽ các nhóm mà mang đấy là cái trường hợp liên hợp thứ nhất. Axit của cái phân tử khi mà xuất hiện cái nhóm siêu liên hợp này, đối với ba trường hợp này ấy, thì trường hợp cuối cùng ở đây thì có nhiều cái yếu tố đẩy E nhất, có nhiều nhóm đẩy E nhất. Thị Minh Anh.
có ạ.
Thị Mai Kha. Đúng ạ. Đây là bạn Nguyễn Thị Anh Thư chú ý, gốc tự do này nó là một cái tiểu phân.
Đấy là gốc đẩy E cho nên là nó sẽ mật độ E nó sẽ dịch chuyển.
Tiếp theo là em cho nó tác dụng với
Thành CH3COCl.
Em thưa cô là
Rồi em nói đi.
em thưa cô là cái này phải cho tác dụng với lại SOCl2 ạ.
Tức là thêm một bước nữa đúng không?
Vâng.
Tức là đầu tiên là men giấm thì tạo ra là axit acetic đúng không ạ? Đúng không Vương?
Đúng ạ.
Rồi sau em phải cho thêm SOCl2 để tạo ra là chloroaxit ở đây ạ. Đấy thì chloroaxit này thì nó có thể tác dụng với aniline để tạo ra cái hợp chất này. Ở trong đề bài là người ta cho cái gì em nhỉ? Cho benzen và cái gì? Metan và các chất vô cơ đúng không em?
Vâng ạ.
Thế thì ở đây nó có một cái hơi bất cập là chúng ta sử dụng men giấm ở trong trường hợp này phù hợp hay không? Bởi vì men giấm không phải chất vô cơ đâu. Em nhé.
Dạ vâng.
Nên là tốt nhất là em thay bằng cái khác. Bằng chất oxy hóa.
Ạ.
Gì em?
Vâng.
Thì em có thể thay bằng đầu tiên là oxy hóa thành aldehyde là CuO, sau để oxy hóa tiếp. Thì có thể lựa chọn là các cái ví dụ như là thuốc bạc này hoặc là lựa chọn là KMNO4 vân vân. Cái hợp chất có ý kiến gì bổ sung cho bài này ạ? Xin mời các bạn khác ạ. Hoặc là có ai có cách làm khác hoặc là có ý kiến thêm gì có thể mở mic ra nói cho cả lớp được không?
OH ạ, để tạo ra sản phẩm ạ. Với dung dịch HNO3 19% ở nhiệt độ 0 độ C. Sau đó là sẽ thu được vị trí beta và vị trí para và octo của NO2 được không ạ? Sau đó là em tách chương cuốn hơi nước hai cái đấy ra, xong rồi em mới cho tác dụng với CH3OH ạ, để tạo ra sản phẩm ạ.
CH3OH á? Tên là gì nhỉ? Cô tìm bài của em á.
Em gửi ở cái phần đầu tiên nhiệt độ ấy, không biết là em chép bài nhầm hay sao ạ, thì phải là 80 độ C. Sau đó là ở chỗ tác dụng hai cái sản phẩm đấy ạ. Và để tách hai sản phẩm đấy thì như bài hôm trước cô dạy là cho chương cuốn hơi nước ạ. Bởi vì có liên kết hydro nội phân tử, cho nên là cái ở vị trí para nó sẽ có nhiệt độ cao hơn, nhiệt độ sôi cao hơn, cho nên ta dùng chương cuốn hơi nước được. Sau đó là em điều chế CH3OH rồi cho cái NO2 ở vị trí para đấy tác dụng với CH3OH để tạo ra sản phẩm ạ.
Thì cái phản ứng cuối cùng là của em nó là phản ứng Freeland-Graf à?
Vâng.
Thế Freeland-Graf rồi đúng không?
Vâng.
Cái này là hôm mà cô dạy cô cũng nói là cái phenol này nó hay tác dụng với xúc tác ấy. Vì trong cái phản ứng này chúng ta phải dùng xúc tác đúng không ạ? Cho nên là trong cái phần này thì như cô giáo nói là chúng ta sẽ thay thế bằng cách là chúng ta sẽ phải sử dụng các cái alken thay thế thì nó sẽ tốt hơn. Còn nếu không thì chúng ta phải tìm cách chúng ta bảo vệ được cái nhóm OH. Thì cái này xúc tác của phản ứng Freeland-Graf thường là AlCl3. Đấy, thì cái chỗ này nếu như em sử dụng thứ nhất là nếu mà dùng phản ứng Freeland-Graf là phản ứng alkene hóa thì thông thường người ta hay sử dụng là CH3Cl em ạ. Đúng không? Còn không hiểu sao em lại chuyển thành CH3OH để làm gì em nhỉ? Cô cũng chưa hiểu ý của tác giả là như nào, em có thể trình bày được không?
Thực ra thì lúc làm bài thì em không em tại vì muộn quá rồi em nghĩ thế nào em làm như thế cô ạ.
Cái chỗ này nó có một cái vấn đề đó là cái phản ứng alky hóa và phenol này nó người ta hay sử dụng alken thay thế và xúc tác là H cộng thay thế cho các cái phản ứng thay thế cho các cái phản ứng của cái dẫn xuất halogen hơn. Đấy thì bởi vì là nó hay bị nó cái hợp riêng cái phenol này nó hay người hay làm như vậy bởi vì là nó hay tác dụng với hai phút.
Let me pull out an old differential equations text.
Video bên cạnh đấy hoặc là nó sẽ để tạo khi mà nó tách nước như vậy thì nó sẽ tạo ra axit không no, đúng không ạ?
And in these books, you will see a description of what happens when you make ring systems that have uncomfortable bond angles. These strained ring systems can cause functional groups to have very different reactivities. So for example, when you put a double bond into a three-membered ring in this form or in this form, a lot of strain energy gets pent up in that ring. And if you can react those pi systems and release the strain, that can accelerate the rate of a reaction dramatically. So I talked with my students about whether there was precedent for an azide alkyne cycloaddition with a strained ring system. And Nick Agard, who is sitting here in the audience today, went to the library, which used to exist, and he found this amazing precedent. Back in 1961, Georg Wittig, another Nobel laureate in chemistry, reported on the reaction of cyclooctine, the smallest of the stable cycloalkynes, which has 19 kilocalories per mole of strain energy. And when Wittig and Krebs, his coworker, combined cyclooctine with phenylazide, you can see from this statement in German, the reaction proceeded like an explosion to form the corresponding triazole. So here was an azide alkyne cycloaddition with no catalyst that basically proceeds at room temperature without the need to heat it up. So based on that precedent, Nick and then Jeremy Baskin, who's now a professor at Cornell, and he's sitting here in the audience today as well as several other coworkers, went to task to try to develop cyclooctine-based probes for what we called a copper-free click chemistry that we could use in living systems. And to make a long story short, they spent several years building cyclooctines with different kinds of modifications. And what we learned is that the cycloaddition of these cyclooctines with azides can be tuned in terms of reaction kinetics over several orders of magnitude. So we had a lot of control levers on the rates of these cycloadditions. Just to give a few examples, in the upper left is a very simple cyclooctine, which would react with a model azide with a second order rate constant that was kind of on par with the Staudinger ligation. So that was our starting point, but we knew that we needed to enhance the rate by a few orders of magnitude. And so Jeremy Baskin had a big breakthrough in this project when he showed that addition of 2 fluorine atoms next to the triple bond would increase the reactivity almost 100-fold. And this reagent, which we gave the acronym DIFO for a difluorocyclooctine, became one of the first important reagents for imaging sugars in live systems, as I'll show you next. But just to complete the slide a little bit, we could enhance the rate yet another order of magnitude by fusing benzene rings on either side of the cyclooctine, which increases the ring strain even a little bit more. And then the ring could be shrunk down to a 7-membered ring as long as one of the atoms in the ring was a larger atom like sulfur. And one could get yet another boost in the reactivity, where now we have a reagent that's 4,000 times more reactive than our parent compound. So this was a really fun period of time in the lab because we felt like we were returning to our roots as physical organic chemists, doing the kind of structure reactivity correlation studies that were popular 100 years earlier, but that had fallen out of fashion in the new millennium. And yet here we are invoking some of these classic concepts to tune a reaction for biological systems. With these new reagents in hand, Jeremy Baskin and another coworker, Scott Laughlin, took on the challenge of imaging sugars in live animals. And the animal that they focused on was the zebrafish. which is a very popular model organism to study vertebrate biology, with the benefit that zebrafish are translucent. So they're a wonderful organism for fluorescence imaging studies. You can see all of their organs under a microscope as they are alive. And so Jeremy and Scott did experiments where they would take early embryos of zebrafish developing in a dish. They would feed these embryos azido sugars, which would get metabolized. And once those azido sugars appeared on the surfaces of the cells of the animal, they would do the copper-free click chemistry reaction in the live animal with a dye-faux probe, introduce the fluorescent dye, and now we could image the sugars for the first time in a living animal. It was a very dramatic moment in the lab. And they did some experiments where they would pulse these embryos at different time points with different azido sugars and map out sugars appearing in different cell types. So for example, in one experiment, they would do one round of labeling, and then any unreacted azides still present in the fish, they would quench with a reagent that is a phosphine reagent in a classic Staudinger reduction. Then they could pulse the embryo with more azido sugars that would appear at different time points and label them with different colored dyes. And we could create fish that basically had all the colors of the rainbow, where the different colors represented different types of cell surface sugars that appeared during development at different time points. Just to give you a sense of what these images look like, up top is the head of a roughly 5-day-old zebrafish, which has had different sugars labeled at different time points with different colors. And you can see how they're distributed. And below that is basically a round of cell division from an earlier embryo. So here's the intact early embryo. Each arrow is pointing towards cells that are undergoing mitosis. And if you expand the image and take a close-up look at cell division, what you can see is that a single parent cell, I'll wait for it to loop, duplicates its chromosomes, segregates them as the cells divide, and you can see the cell dividing through the portal of the DNA, which there are good tools for imaging, and we used one of those tools here. You could see the cell division through the portal of the proteins, which were also easy to image back then, and the proteins that we're imaging here are located on the membrane of the cell, and now it's dividing into two daughter cells. But for the first time, we could also see cell division through the portal of the cell surface glycans. And that was another first-in-class achievement by these incredibly fearless group of students. And in this image, we were quite struck to see how the glycans build up and concentrate right at the junction as the cell is completing a round of mitosis and contributing to the segregation of these daughter cells in a way that was previously unknown. So after this point of time, the field really took off because it turns out that once you show that you can use chemistry to image sugars in a biological setting where the animals are alive, lots of people came out of the woodwork and asked us if they could use our reagents to image all kinds of other kinds of molecules that were also difficult to study. And so here's a pie chart on the cell showing roughly the breakdown of all the different types of biological molecules. And over the course of 4 or 5 years, and this brings us up to about 2010, there were groups that showed that they could use the azide as what we call now a chemical reporter for virtually every type of molecule on the pie chart. They could put azides into amino acid side chains and label proteins, and use bioorthogonal chemistry to image those proteins in living systems. They could put azides into nucleosides and label the oligonucleotides and use bioorthogonal chemistry to image those molecules. They could put azides into lipids and track those lipids around in living systems using bioorthogonal chemistry. So the applications of this chemistry in the research setting expanded quite rapidly into lots of different areas of biology. Another big development around that time was expansion of the toolkit of reactions that are available for bioorthogonal chemistry. And so we started our work with the azide And the cyclooctane partner became a really good combination for chemistry and living systems. But later, other groups discovered that tetrazenes can be bioorthogonal reagents reacting with strained alkenes like this cyclooctene or this cyclopropene. And now that toolkit continues to grow. And I would put the challenge out to students in the audience here. If you come up with a new idea for a new bioorthogonal chemistry, this could have tremendous impact. Because while we now have a handful at our disposal, there's always a need for more options in this space. It's a great challenge for someone who's starting out their career in organic chemistry. Also, the applications expanded. So 20 years ago, we developed these chemistries as tools for basic research in the laboratory setting. But over the last 10 years or so, there's been considerable activity in the translation of this chemistry for new medicines and new diagnostic strategies for human disease. And so I thought I would mention that there are now a number of biotechnology companies that use bioorthogonal chemistry in a variety of settings. A very popular application is for making antibody drug conjugates. And these are important medicines for the treatment of cancer. As well, there are companies that are using bioorthogonal chemistry to make new vaccine conjugates. And some of these are in, right now, a phase 2 clinical trial. And then I have a company, as a disclosure, which uses bioorthogonal chemistry to make targeted degraders of extracellular molecules. And I'm very excited to be an advisor of a company called Shasky, which is performing bioorthogonal chemistry in the body of cancer patients as a mechanism to target chemotherapies very selectively to the tumor tissue and to keep those chemotherapies away from healthy tissues. So it's a very exciting time for me to see how the chemistry came from a very modest idea in the research lab aimed at imaging sugar molecules into a very generally applicable tool which people are using to make new types of medicines. Finally, I want to make one point which I think is important because despite all of these exciting applications, I still think the most important lesson from the story of bioorthogonal chemistry has to do with the importance of curiosity-driven basic science. And those of us who are scientists are familiar with what people often describe as a tension between basic science and applied science. And funding agencies go through various phases of interest in applied science versus basic science. But bioorthogonal chemistry tells the story of how important curiosity-driven basic science can be in order to realize these applications. Because I'm pretty certain that the chemists on whose shoulders we built bioorthogonal chemistry, and by that I mean Hermann Staudinger, Rolf Huisken, Georg Wittig, when these chemists reported on the curious reactivity of azides, phosphines, and alkynes, they would never have foreseen that someday I would stand here and show you fluorescent zebrafish. They couldn't have known that. They made observations about exotic molecules and they published them purely based on curiosity. And here we are 100 years later making use of these chemistries in ways that we hope will benefit human health. So in order to get to fluorescent zebrafish and imaging of sugars, to imaging of tumors in human cancer patients, and to making new medicines, you need to have that foundation established by the basic science. So with that, let me thank the bodies of people that basically made this moment possible. First and foremost, the Royal Swedish Academy of Sciences and the Chemistry Nobel Committee. Thank you for changing my life. The Nobel Foundation for the amazing festivities of this week, which are just ramping up. My institution of origin, UC Berkeley, which we often call Cal. This is where all the work was done that I presented today. So I owe a great debt of gratitude to the students and to the faculty at Cal that gave me my big break and that did all this beautiful work. And my current institution, Stanford, is very supportive and is helping me try to translate some of these reactions into human medicines. And then finally, the people who financially supported this work from the very beginning, which is the National Institutes of Health and the Howard Hughes Medical Institute. The most important slide of all is this one, because none of this happens without the intrepid students and postdocs that occupy the academic laboratory. And this is my current group at Stanford. This is how we had group meetings during COVID. We had to rent a drone to get this picture. And after the Nobel Prize announcement, my students organized a Zoom party. And these are all the faces that we could get on Zoom on short notice from all over the world, and some of whom I'm presenting this work to today. Thank you very much for the work that you did leading to this moment, sitting there in the second row. My family is here. Thank you very much for all the support you have given me since my moment of birth. And thank you very much to this audience for coming today. Thank you.
I'm Morten Meldal. He is a native of Denmark and was born in Copenhagen in 1954. In 1986, he was awarded a PhD from DTU, the Technical University of Denmark, located in Lyngby, just a bit north of Copenhagen. And he's now active as professor at University of Copenhagen. So by that, please join me in welcoming Dr. Meldal up on stage.
Dear colleagues, friends, Dear colleagues, friends, ladies and gentlemen, I write A Leap from Shoulders of Giants, and I would like to start in reverse order of the talk we just heard by thanking all the giants that are behind the work that I'm going to talk about today. So my heroes, some of them, Klaus Bock here in the audience, together with Wüthrich on NMR, structural studies on carbohydrates and peptides, Hüsken and Woodward and other people that are doing cycloaddition reactions, Prigogine, Bach and Gray on quantum chemistry, Paulsen, Lemieux, and I think that we should also mention Shepard and Merrifield on the peptides and carbohydrates, and then Furka and Lam and many more on combinatorial chemistry. These are really the people that we built on in our research. So before I dive into the lecture, I would like to thank also the Nobel Foundation and the Nobel Prize Committee of Chemistry, as well as the Royal Swedish Academy of Sciences for this absolutely unique event. And I'm very appreciative and very thankful for being able to stand here today and very proud to share this moment with Caroline Bertozzi and Barry Sharpless. I also thank all the colleagues at Carlsberg Laboratory and University of Copenhagen for all the things that they have done during the years in discovery, development, and application of this click chemistry. And finally, I want to express my thanks and love for my family who have supported me over the many years of science, and in particular my wife, Phaedra Saint-Hilaire, my son, Ajani, who is here today and is celebrating his birthday, my daughter, Anna, and Anna's mother, which is not here today, Sandra Davoglio. Finally, I would like to express the thanks to my late parents, who would be so proud to be here. And my perception of nature, and in particular the underlying chemistry, is a testimony to me of the absolute beauty and the complexity of the nature and our very existence. So nature has been with me since early childhood, and I'm very grateful to my parents for dragging me through forests and making collections of rocks, butterflies, insects, and so on, so that they actually spurred my interest for science. And later in life, I chose science as a subject for my education. And at a certain point, I had to choose between chemistry, physics, and computational sciences. And I chose, fortunately, chemistry, because today, to me, chemistry is everything. There's nothing around us that is not chemistry. You are chemistry, I'm chemistry. All our surroundings are chemistry. And in fact, we will need chemistry very much if we have to solve all the challenges that are ahead of us. The global challenges will need chemistry solutions, and we need our young people to learn chemistry from the very point. So from very young, we need to go through this process and learn about chemistry.
Good job breaking through in the end game with strong.
After
they made one of the top moves in this move.
Study,
they made one of the top moves in this, developing the
and
peptides. This move,
and I spent some time at the university, and then I ended up at Carlsberg Laboratory.
Your best move was to win material.
And at Carlsberg Laboratory, I combined glycopeptides or peptides and glycans into glycopeptides in order to start a study glycoprotein.
A solid recapture by White, bringing a new piece into play.
And during this process,
this move, you moved quickly, missing the only good. They exchanged pieces of equal value. You moved quickly. This move protects
and the reason was that
you permitted
there
the
was
opponent,
a great need for quantitative chemical reactions at that point. So we had a great focus on QCT
you had a
reactions,
chance to play something better.
At the time.
This is the only logical capture. They played this move doesn't hurt you much. They played the strongest move available.
And for those who don't know what combinatorial chemistry is, I would like to give a short explanation.
Your better option was
we have
a clear
some
recapture.
Small plastic beads that we use for combinatorial chemistry, and they can contain different starting points. As you can see, they're orange and green, and
your opponent has to watch that.
And
pushing
now we can couple reagents with quantitative chemical reactions to these beads and form one combination. We can then mix these beads up into a complete mixture, and then we can redistribute them into these wells and couple a second reagent. Now, each of these small plastic beads is a container for biological assays, as well as they contain only one chemical compound. So if we have 3 Lego blocks and 3 steps of reaction here, we'll get 27 compounds, or 3 to the third. If we have 5 steps and 5 compounds, we get 3,000 compounds, or 5 to the fifth. If we have 20 compounds and 5 steps, we'll get 3.2 million compounds. So you can see this is an exponentially growing process of generating compounds at very large numbers. And we therefore needed these very perfect reactions in order to do that. So during this time, this is showing how we can do this in practice. The beads are in here. We do the coupling reactions, or mixing them, and we prepare them for another coupling reaction, and we take around this cycle many times. At the end, we can take these beads into a biological assay with enzymes or binding biomolecules, and then we can identify the active compounds on those beads that are showing a sign of activity. So this is, from the combinatorial chemistry side, very important that we can identify those compounds here, like needles in a haystack, and we are using mass spectrometry to do that work. but this is another story. Because here at Carlsberg Laboratory, we did the discovery and the multifunctional molecules by click that I will describe. Click reactions, what is it actually? The click reaction. And then I have some applications I would like to present to you. During this very active period from 97, a lot of people were working in the lab on establishing N-acylaminium reactions, amination reactions, and also we tried to make ketoalkynes for branching peptides and making Diels-Alder reactions on peptides. And this here is a method for peptide synthesis that we developed where we can synthesize extremely difficult peptides that cannot be synthesized in any other way. It's based on the A site as a protecting group for amines and the acid chlorides for coupling. Very high activity of these compounds in the coupling reactions. So one of our very skilled PhD student, Christian Tornø, who is here in the audience, had a project in which he had to take alkynes, make the copper acetylenes, and then react those with the acid chloride so we get these acyl alkynes. That did not happen in his reactions. He fought a brave battle in order to make this reaction work over several months. Nothing would work, and the reason was that this irritating byproduct was formed all the time. It was such a pain, but when we looked in detail into this irritating byproduct, we found that this was actually the triazole, and it was formed quantitatively even in the presence of this very reactive acid chloride. And you can see there's no starting material left, and everything is converted in a short period of time. So Christian investigated the compatibility of this reaction with all the peptide functionality that you have in proteins. And he presented this work at the APS in San Diego here. So thank you very much, Christian, for your work. Carolyn alluded to a universe. I'll just talk about a toolbox that pharmaceutical industry is using with all these molecules that we make in pharmaceutical industry and the many problems that are associated with solvents, with excess reagents and so on in this toolbox to form the well-known drugs that we are taking today. Also in material sciences, you have these kind, for example in polymer synthesis, you have these kind of tools that are not very clean and not very green chemistries. And then you have a very small toolbox today, as introduced by Carolyn already, which contain the QARC reaction and Carolyn's two reactions that you see here. This little toolbox contain, as you also saw from Carolyn's slides, only 3 reactions. And it's 20 years ago we started this research, which means that it's extremely difficult to find reactions like this. So we have this little toolbox that's completely orthogonal to the chemistry going on out here. And that means that we can take things from this big toolbox and combine it like molecular Lego. So I would like to show you what we can do with click chemistry in a putative example where we have formed this guy here. He is good to kill cells from the inside by apoptosis. Then we have this monoclonal antibody that can locate tumor tissue, and we have a cell-penetrating peptide that can take the other guys with them into the cell if they are connected. So now we take these 3 guys and we put them into a solution of copper-1 ions. And once they're in there, they find each other on the surface of the copper and they couple and make the triazole. So we can do that even in with completely deprotected proteins and in water solutions. This is quite unique. And we get this guy here, and now this guy, he can find a cancer cell like this one, and he sits on the cancer cell for a while, and the cell-penetrating peptide does the work, carry them inside, and now the apoptosis can work, and if we're lucky, the cell is dead. So click is good, and click is green. So click can be carried out in water, and click doesn't lose any of its material. If we are clicking these two guys together, you can see we can find all the atoms in the product. So atom economy is very important here. So this is a kind of reaction that you would really like in order to combine two large parts in a new pharmaceutical. But what is really click? We start out with an alkyne, and this is a big bag of electrons. It's full of electrons in orbitals or balloons or whatever you would call them, here sitting on the surface of the alkyne. That's negative charge. It would love to see a copper 1 atom and bind it here to this orbital. That acidifies the alkyne out here, so the copper atom takes the place of the proton on the end of the alkyne. That's still not reactive enough, so we take another copper atom and take it in. It comes in there, sits on the electrons, and it peels off the electrons from the alkyne. So we get a positively charged alkyne, so to speak, with empty orbitals that are looking for electrons. And where can we get the electrons from? Well, one place we can get them from, which fits almost exactly with the faces of these balloons here, is the A side, and here comes the A side, and it fits exactly to form what we call a metallocycle. The metallocycle that lose first one copper atom to form a triazole and a second copper atom